All right, so it is 2 o'clock, so we will get started. Let me pull up the PowerPoint. Not showing up for some reason, so let me just reopen that and try again. Okay, share screen. Weird. So I can't open up the PowerPoint like I normally do, so we're just going to share my whole screen then. That should be fine. All right. Let me get to duplicate and move this out of the way. Okay, so we were talking about GMOs. We were talking about plasmids on Friday. And then we talked about PCR at the very end. And so we only have one more slide to go on this PowerPoint, then we're going to go to today's. And the last thing we need to talk about PCR are just some primer characteristics and doing some thought experiments on what happens if you make a mistake in PCR. So some ideal primer characteristics. So I can just answer this one for us. So when you are making a primer for PCR, generally what you want to do is have your primer be about 18 to 20 nucleotides in length. Your melting temperature should be roughly 52 to 58 degrees centigrade, and it should be roughly 40 to 60% content GCs. And when you're doing this, what you do is that you actually go to a website and you can make your primer in a website, and it will check for all of these characteristics. It will also check to see if your primer will form any structures within itself, because if it can bind to itself, that lowers your PCR efficiency. So you just type in like PCR primer into Google, making PCR primer. And there's a lot of websites out there that will do calculations for you. And then you just order these primers from a company, and you are good to go. So let's think about what happens. GC are the bases, G and C. That's what GC stands for. And by the way, since I'm sharing my screen, if you send me a private message, everybody will probably be able to see that thing. So just FYI, if you want to talk to me privately, this session's probably not the best one to do it. So just send me an email and I will get whatever's happening to my computer, I'll get that worked out by Friday, why I can't share just the PowerPoint anymore. All right, so what are the possible outcomes of PCR in which we remove a primer from a reaction mixture? So answering A, B, C, and D here will really help you understand how PCR works. So here's my DNA. Remember, my primers are supposed to flank what I want to duplicate. So I have one on each strand. Well, if I only have one primer, then I'm only going to duplicate one strand over and over and over and over again. So basically, I'll only have a lot of copies of a single strand of DNA, and I won't have the other copy that I need. So I won't be able to properly make my sequence. So that's why you need both primers. Both primers copies both strands of DNA. And they also will dictate, what section of DNA will be copied over and over and over again. So if you only have one primer, you can't do that. What happens if your primer can bind to multiple sites? Well, you're going to get strands that are multiple lengths. So ideally, you want your primer to only bind to one location. But if I bind to multiple locations, you're going to get DNA of different lengths. And again, you're not going to amplify the sequence that you want. So when you're designing a primer, it is good that it can only bind to one place, so you can amplify only one section. What happens if you have a single strand break in your DNA? So what if you want to replicate DNA that looks like that, where we have a break? And this one is actually okay. It does not matter if you have a single stranded break. Because let's take a look at why that is. Let's say we have a primer here and we have a primer here. The bottom primer is going from right to left. The top primer is going to left to right. After one round of PCR, you would get this back. You would make a duplicate of where that break was. You would still have a single strand DNA that corresponds to that. However, your bottom strand would be able to make a whole sequence of DNA no problem, right? You'd copy the bottom strand, and you also have the top strand left over from your from the parent. So that's round one. Round two, the primer starts here, primer starts here. Now you're good to go. You have a copy of your DNA that has no breaks in it. Now you can duplicate your DNA like normal. So if there's a single strand break in your DNA sequence, that's not really an issue. What is an issue is when you have a double strand break. If you have a double strand break, your DNA is cut in both strands at the same place, you can never successfully replicate that DNA without fixing that break first. So let's take a look at that. So let's just imagine right there, my DNA was broken. Right? And so my primer's attached, I do PCR. What I'm left over with is something that looks like this. One small portion of DNA replicated, one larger portion replicated, but I can never get back the sections that are broken away. I don't know what's there, so I can't replicate it. So with a double strand break, I will never be able to do PCR successfully. Single strand break, you can though. So is there any questions about PCR, primers, or anything at all before we move on to today's material? Clear that. Okay, so let me get the PowerPoint up for today's work. Oops. All right. Share that. Okay. So our previous PowerPoints, our previous lectures were covering our DNA. Now we're going to get into our next polymer. That would be the amino acids. So what you need to know for the amino acids, and I believe my the videos I posted online already, have some ways for you to remember the amino acids. What you need to do for me is you need to know the full name of the amino acid, the 3-letter code for the amino acid, the 1-letter code for the amino acid, what amino acid looks like, the side chain, if the side chain has a pKa, and the N-terminus and C-terminus are set. So the N-terminus pKa of all amino acids are 9.4. Usually I just say 9. C-terminus I usually say is 2. So for this, a lot of memorization. If you have a smartphone, right, and go into like any of your app stores, I'm sure you can find flashcards for memorizing amino acids because it's, part of the MCAT. Every biochemistry student has to do this. I'm sure people have material out there. If you're more old school, you can buy flashcards and write them down. That's what I did as a student. I made my own flashcards. But basically, that's what I want from you. There's 20 amino acids. Know the full name, 3-letter code, 1-letter code, and what it looks like. And if the side chain is ionizable, what's the pKa? Now, to the left here, we have our general amino acid structure. And amino acids are what are called Zwitterions. Zwei is German for 2, ion. So amino acids under normal pH 7 conditions always have 2 charges. They always have a charge from this nitrogen and a charge from this carboxylic acid. What I just drew right here is the backbone. That's what we call it. It's called a backbone. Every amino acid, it's going to have the same backbone. It's going to have NH3, and then it'll have a carbon. And I wrote an alpha because this is called the carbon alpha, right? Carbon alpha. And then it's going to have a carboxylic acid. This R group, anything that's coming off the carbon alpha, is called a side chain. And we have 20 standard amino acids, and they have 20 different side chains. The simplest being a hydrogen, the most complicated being a double ring structure. So basically, know all of those amino acids, know the side chains. Some people might tell you there are 21 amino acids. I'm going to say there's only 20. All right. So when amino acids combine to make proteins, what happens? So since I'm sharing my whole screen, let me move this up a little bit. What happens is that 2 amino acids will come together and they'll make water. And you form what's called a peptide bond. So you're going to lose 1 oxygen, 2 hydrogens, and make water. And that's called a dehydration reaction. Because you're removing water from your structure. You're dehydrating it. And you're making this peptide bond between the carbon and the nitrogen. And to break this, to reverse it, you need a lysis, and it's called hydrolysis. So using water to cut something, to go the reverse, to take a peptide bond and cut it, you need water and you need to cut it. Whenever you see the word lyse, that means to cut. So hydrolysis is water cutting. So those are the reactions, the name of the reactions for combining amino acids and cutting amino acids. Now here at the bottom, I have a simple peptide. So when you hear the word peptide, Peptide just means amino acids connected together with peptide bonds. Proteins are peptides. The words are kind of interchangeable in that all proteins have peptides, but maybe not all peptides are proteins, right? It's kind of debatable where you say a peptide becomes a protein, and it's a lot of semantics. So I wouldn't get too hung up on it, but basically, if a peptide's like over 50, 60 amino acids and it can fold, you usually call it a protein at that point, but you can still call it a peptide. That's fine. Now here, we're looking at 4 amino acids. The red parts are the side chains. That's what gives them their distinction. So we have alanine, tyrosine, aspartic acid, and glycine. Now, if we consider the free amino acid, tyrosine, which, I cannot draw, which is shown here, so I'm going to draw that. So there's tyrosine, the amino acid tyrosine. It's barely soluble in water, barely soluble. So do you think if I make a peptide chain of tyrosine, would that be more soluble or less soluble than a single tyrosine? And tell me why. Remember, if you send me a private message for this class, people will be able to see it. But please, if you have an idea of, do you think tyrosine would be more or less soluble in a peptide, share your logic with us. Remember, a peptide is just connecting amino acids via a peptide bond. Do you think that will make, if it's just tyrosine, do you think that'll make it more soluble or less soluble? More soluble, because the N-terminus and C-terminus are still charged, so it wouldn't matter if tyrosine is hydrophobic. Okay. Any other suggestions? So we have one thought there. So we have a vote for more soluble. Tyrosine by itself is less soluble. And so connecting tyrosines will make them more soluble. Okay. Anyone agree or disagree? What are the thoughts out there? Or you can just answer, I have no idea. I suppose that will work too. To know where people are at. Charge mean hydrophilic, right? So charge is hydrophilic, but if you're hydrophilic, you don't have to be charged. Charged means you have a net positive or net negative overall charge, so you are hydrophilic by definition, but you can be hydrophilic in neutral as well. No one else wants to speak up? No one else has any ideas? I think the peptide would be more soluble. Would it be less soluble because there is more hydrophobicity? Okay, so we have two more soluble, one less soluble. More soluble. And don't ever be afraid of being wrong. If you were right every single time that anyone ever asked you a question in college, there would be no point of you going to college because you know everything. More soluble, less soluble, only because you all say less. Herd mentality, I like it. Less soluble. Because the peptide includes the N and C terminus, so it's just not T. More soluble. Okay. The answer is less soluble. The reason why it's less soluble is that if we look at the structure of tyrosine, the amino acid, it has some hydrophilic parts, right? It has a positively charged N terminus, has a negatively charged C terminus, and it has an OH group. So those are all hydrophilic. They like to interact with water. It also has this giant carbon ring, this phenol group. The carbons, the water does not want to interact with the carbons. So what happens as you add more and more tyrosine, and I'll draw it down here. As you add more tyrosines, sorry for my awful drawings. You are adding a lot of rings, a lot of more or less benzene rings. And look at the backbone though. You only have one charge N terminus and one charge C terminus. The rest, sure, they can hydrogen bond, but they're no longer charged. So on the peptide bond, right? When you form a peptide bond, the C and the N lose their charge. They're still polar, but they're not charged anymore. So when you connect tyrosines together, you're adding these big, bulky, hydrophobic groups over and over and over again. You're not really balancing them out with hydrophilic groups all that much. So tyrosine by itself, barely soluble, barely soluble. You add more benzene rings to that, it's going to crash out a solution. All these rings are going to interact. They're going to be pushed together by water, and you'll just have a big solid blob on the bottom of your test tube. So questions about that idea? Yes, all tyrosines, like you put like 200 tyrosines in a row, they are just going to glob together. They will not, water will not want to interact with that molecule. Are you trying to say I just don't get it? Or I just don't it? It's so funny. A funny joke. Yeah, well, the way to remember it, all right, is just think, does water want to interact with this? Water does not want to interact with any of these rings. You add more and more and more and more and more rings, water is just going to push these together, hydrophobic effect. Yeah. So how else to explain this? So let's think of another molecule. Let's think of leucine, right? Leucine by itself no, that's isoleucine. I just drew isoleucine. By itself, isoleucine again, you have a lot of carbons and two charge groups, a positive charge and a negative charge. Those charge groups can interact with water. They can hydrogen bond. This big old side chain can't. So if you drop one isoleucine amino acid in the water, sure, maybe it would be a little bit soluble because you will interact with these charge groups. As you connect amino acids together in a peptide bond, when you make that connection, the N and C are no longer charged, but you keep adding these big bulky side chains. If the side chain is hydrophobic, water won't want to interact with it, and so it's going to push that amino acid away. And if it's really big, what's going to happen is that water will push all the hydrophobic side chains together, and it will eventually crash out. But the basics of how a protein works, how a protein folds, is that you have some hydrophobic amino acids, some hydrophilic. Water will push the hydrophobic amino acids together and interact with the hydrophilic. So the really thing to understand here is the more hydrophobic groups you add to a protein, the less it will dissolve in water, less likely it is, because water won't want to interact with anything it can't hydrogen bond with, right? Rings are so hydrophobic, doesn't matter. Yeah, the idea is the more hydrophobic rings you add, you're only going to have two charged termini, one N and one C. So if you only have one hydrophobic ring, that's okay. It will still dissolve. You add 100 hydrophobic rings and still only have one charge N and one charge C, it's not going to want to interact with that molecule. The side chains interact, they do. But that little OH on top of the ring, that's still tiny. That's very tiny compared to the big ring. That water is not going to want to interact with those rings. So it's going to push them, it's going to try to push them together, the hydrophobic effect. All right. Hopefully that was a little bit clearer after that discussion. So one of the things we need to do is we need to learn how to draw peptides. So what I would suggest, first starting out, and if we're in class, I would give you this, but everybody should have like a Google image of amino acid side chains, or just Google amino acid side chains or Google amino acids, because we're going to learn how to draw peptides. For the sake of time, I'm going to draw this one for us because just to get it, show you what it looks like. But I do recommend that you come back to this problem and look at it without the answer, or just create words and try to draw what the peptide looks like. So we have the peptide tapes: threonine, alanine, proline, glutamic acid. I was going to say sulfur. Why can't I? Serine. There we go. Serine. All right. So every protein always starts with an NH3. Let me go back to my nice little dark blue. So when drawing a peptide, you always start with a charged NH3 group. This is called the N-terminus, right? How long do we have to learn all this? Until test 1. I forget off the top of my head when test 1 is, but you will see questions on it on test 1. Yeah, I mean, it's not hard. There's tricks. But yeah, let me draw this. While I draw it, so the first thing we do is N-terminus carbon alpha. It seems hard. There's only 20. Okay. Let's go over some tricks. Instead of drawing tapes, I'm going to go over some tricks first. Okay. So every amino acid has that backbone, right? The simplest, which is easy to remember, is glycine. Glycine just has the side chain of an H. Oh, that's horrible. H. So glycine H. G, glycine. And didn't you all pass organic chemistry? Isn't there like a crap ton of memorization in organic chemistry? This is only 20. It's not that bad. Okay, next one, alanine. That's a methyl group, all right? What happens if I add a OH to that methyl group? Then you get serine. CH2. What if instead of an O, I want an S? That's cysteine. So the way I do this is I start with the simplest and just start building off there. That's what helped me. Leucine looks like an L, the side chain. Isoleucine is like leucine, but it's different because it's called iso. So this is a rite of passage for every biochemistry student. Every biochemistry student in the nation has to learn all 20 amino acids, their names, their side chains, their pKa's. Any of you wanting to take the MCAT or the farm, whatever that's called, you're gonna have this on there, right? When we wanna talk about proteins, I need you guys and gals to know if I say alanine is in our binding pocket, what does alanine look like? What structure does it have? If you put a little bit of time in it, I promise you can memorize this. Do we alternate the size that we draw the side chain on? You don't have to. I draw it all on one side. Does spelling count? Yes. Oh, wait. Oh, I see what you mean. If you're close, I'll count it. So if you misspell the 3-letter word, the 3-letter code for serine, right? If you give me SAR for serine, that's not correct. So I would count spelling there. But if you said serine is called sarine, I would probably be more forgiving, because the 3-letter code actually means you need the exact 3 letters there. If you give me the full name and it's not quite there, I'll give you some leeway. So for this problem, since we already did some drawing here, basically what I will be looking for, you need to start with a charge N-terminus, carbon alpha, and then I'm just going to draw R. So the R are the side chains. Then it goes C double bond O. Then, so this is amino acid 1. Your next amino acid goes NH, C alpha, C double bond O. Amino acid 2. Next side chain. Amino acid 3, NH, carbon alpha, C double bond O. And you don't have to put the hydrogen up and the C double bond O down like I do. That's just the way I draw them. Amino acid 3. Next, and you draw the side chain. Okay, amino acid 4. What's the backbone going to be? Well, NH, carbon alpha, C double bond O, R. Amino acid 4. Okay, amino acid 5. So this would be TAPES. Okay, amino acid 5, NH, carbon alpha. However, we're at the end, so now we get a negative charge. And where I put the R's, you just want to put the backbone. So that's or sorry, the side chains. The backbone's always the same. The side chains are what are different for us. Yes, this class is more interesting than organic. Do you have to put the brackets? No, I suggest you don't put the brackets because the brackets are just my way of showing you where one amino acid starts, one amino acid ends. In reality, you would never put the brackets. So you will probably never see me draw those brackets again for peptides. I'm just doing that so we can remember where one amino acid starts and where it finishes. One thing I do want to, let me clear all this nonsense. One thing I do want to point out, proline, P, is special. So, and, okay, so these squigglies mean I have amino acids to the left and amino acids to the right. Proline, what proline does, and you'll see this when you look at the structures, is that proline will bind back on its own backbone, and you form a covalent bond between nitrogen and the side chains. So that's not double bonds. That's just, let me undo that. So nitrogen or proline will bind back on its own nitrogen. So there is no hydrogen for proline. Proline is the only amino acid who has a different backbone because it binds on itself. And the only difference is there's no hydrogen on the amine group. There's no hydrogen on the nitrogen of the backbone. Proline is our one exception to the backbone, always looking the same. The backbone is same, only C alpha. C alpha is always the same. The only thing that's different in any amino acids is what's coming off the C alpha, which we call the R group. The R group is what we call the side chain. There are 20 different side chains you need to know for our amino acids. And each side chain gives the amino acid a different property. All right. We'll probably just have to recognize them in a real, if this was non-COVID times, you better believe I'd ask you to draw them. During COVID times, I'm probably just going to ask you to recognize them. So you got something good from being all online class. It'll be a little easier. It's easier to recognize than to draw them. Drawing helps you memorize them better. That's why I do it. But limitations. How do you draw the end term? Sure. So carbon alpha, that's the C terminus. That's the end. The end terminus is the start. And just like how we read DNA 5' to 3', you always read peptides end terminus to C terminus. They're always created end terminus to C terminus. And you have nonsense right there. Okay. So yeah, practice, practice, practice. If you want to like have some good practice, draw your name, right? Take your name and draw it out as a peptide, hang it up in your room to show anybody who comes over how cool you are, that you can draw your name and amino acids. I mean, I would be impressed if I went over to someone's house and they had their name drawn in amino acids. That's just me, but do we need to label beta, gamma carbons? No, you don't even need to label the alpha carbon. I just do that out of habit, and it's called the alpha carbon, but no, you don't need to do that. So when you have proline in a peptide, live, laugh, love. You cannot have live, laugh, love. There is no U peptide or U amino acid. Can I draw an example of proline versus other amino acids? In between two other amino acids? Sure. So let's do, let's make it easy on me. Whoa. Let's do G, P, A. Did not mean to make that GPA. I just wanted to pick the easiest. Anyways, GPA. Carbon. Glycine is just a hydrogen, so hydrogen. Glycine done. Next amino acid, proline. Proline done. So that's G, that's P. That's alanine, A, GPA. So proline doesn't have this hydrogen right there. That's the only difference. It binds back on itself. What would be the best way to prepare for the exam? I mean, what do we study? Lecture notes and so lecture notes, study guides, previous tests are what I would do. That's how I would study. Interesting tattoo, that would be a long tattoo. I don't like proline. Proline is special. Does it always have that weird pentagon shape on? Yeah, because that's carbon, carbon, carbon. Those are all carbons, right? Now, I remember from organic chemistry, if I just draw all in lines, every time I make a point, that's a carbon. So yeah, you just have carbon binding on it, binding back on itself. Proline is, we'll talk about why proline's special when we talk about actual proteins. Proline has its purposes. All right. And if you ever go to the store, you can buy proline in powdered form and pill form and eat it if you wanted to. Okay, let's move on now to pKa's and PIs. So this next section, historically, has been the one like how everybody just revolted, like in the last one when I told you to memorize 20 amino acids. This section is where historically I get the revolt. So maybe I'll get 2 revolts this year. Okay, so amino acids are ionizable. That's why they get the name acid, right? You have one part that can act like an acid. Sometimes they have 3 ionizable groups. So every amino acid has an N-terminus and a C-terminus. As we just saw, those are charged. And if you're ionizable, that means you can become charged. Some amino acids, and this is just something, again, and I'll write them down. Some amino acids have side chains that are charged. So at a pH of 7, your positive amino acids are R, K, H. And at a pH of 7, your negative amino acids are D and E. You might want to create like little acronyms to remember that. But yeah, anytime we talk about charge, we're going to talk about at pH 7 for the most part. Now, at different pHs, our amino acids can take on different charges. And so at low pHs, at pH of 1, everything becomes protonated. So anything that was negative becomes neutral. At high pHs, everything becomes deprotonated. So anything that was positively charged becomes neutral. However, at the extremes, we need to do a little bit of math to determine what is the charge. And really, the only formula we need for that, the only thing we care about all right now is what's called the isoelectric point, or PI. PI is the pH at which an amino acid has no charge, right? You can use PIs for proteins if you wanted to as well. They're actually used quite a lot for proteins, but right now we're just talking about amino acids. And the formula for PI is that if your amino acid is neutral at a pH of 7, the PI is simply the pK of the N-terminus, which we're going to say is 9, plus the pK of the C-terminus, which is 2. So you do 9 plus 2 multiplied by 1/2 to get the PI of neutral amino acids, or at what pH are that amino acid going to be neutral. And I'm using 9 and 2 here. Every amino acid has a different pKi and a pKj. For the N and C terminus, we just have to remember N and 2. So positive amino acids are RKH, negative amino acids are DE. Or PKI or PKJ are just divided by 2. So for this part, we will do practice problems. That's me just saying it, you're not probably going to get it. But I'll show you what the math is. So let's say that we have a protein, N terminus, pKa, or pKa is 9, C terminus is 2. So what pH is it neutral at? So 9 plus 2 divided by 2, or 11 divided by 2, this amino acid is neutral at a pH of 5.5. That's when the amino acid has no charge. It is 100% completely neutral at a pH of 5.5. For negative amino acids, we take the C terminus. Again, we're just for simplicity's sake going to say the pKa of all C terminus are 2. And we're going to say the side chain. And every amino acid, every negative amino acid has a different pKa for the side chain, or pKj. So let's say that is 3.7 for our argument's sake, right? So to calculate pi for this, it is equal to 1/2 multiplied by pKi, the C terminus, which is 2, plus the pKj for aspartic acid, D, that is 3.7. So it is 5.7 divided by 2, which is 2.85. That is when this amino acid is completely neutral. At a pH of 2.85, this amino acid with those pKa's are neutral. Positive amino acids, all you do is you take the N terminus and the side chain. So for amino acids, we can determine when it's going to be electrically neutral. Proteins, it's way harder. So you either do experiments, or there are some really complicated calculations that are done by computers that can estimate at this pH, your protein should be uncharged. On the PowerPoint, it says PK1 and PK2. Let me go back. So, in the top. So the I and the J, yeah, they can be 1 and 2. It really is, if you want to call that like PK1 plus PK2, that's fine. All that, or I and J, that's fine. All that matters is that you know what's going into these calculations. For neutral amino acids, it's the N terminus and the C terminus. For negative amino acids, it's the C terminus and the side chain. For positive amino acids, it's the N terminus and the side chain. So if it's neutral, use both termini. If it's negative, use the C terminus, which is negative, plus the side chain. If it's positive, use the N terminus, which is positively charged, plus the side chain to do these calculations. So that's all this slide is really saying. Any other questions about that? -1 is only used on 2 amino acids. +1 is only used on 3 amino acids. Basically, yeah. There are some other exceptions if you go to different pHs, but 99.9% of the time, yeah, that's true. How do we get numbers from the graph? You get the side chain numbers through memorization. From those, you can Google it. They're all going to be the same. I can post up. Yeah, that's what I'll do. I'll post the handout I usually give that has all these numbers. But the C and N term list just always say N is 9 and C is 2. They all have different numbers, but they're basically the same thing. You should trust Google. If you Google amino acid pKa's, you're gonna get the right thing, because who's gonna put up the wrong information for that just to trick people and for it to get enough clicks for it to be the #1 search result, right? But anyways, I'll post up the one I usually hand out. Fake scientists exist. They do, but that would be really weird for them to put that up there. So it's a PI where the average pKa is neutral. The PI is the pH where that amino acid is neutral. It's the pH where the amino acid has no charge. Now, we'll call it here. This next section, question 2, is what usually everybody screams at me for. So this will probably take most of the time on Friday. I'm guessing that'll take half an hour. So we'll see where we are on Friday. I'm gonna keep the quiz or not the quiz, sorry, wrong class. I'm gonna keep the reading guide up for Friday, but depending on how fast we go, I might move the reading guide that's due on Monday and move that back a day. But I'll let you know on Friday where we are on that. Otherwise, I will put up a homework for what we covered today. And then on Friday, I will teach you how to get the overall charge for peptides at different pHs. And I'll also post the handouts I have, all these side chains of the amino acids with all their pKa. So you have a nice little reference base for you to start memorizing. Yeah, so welcome to biochemistry, everybody. We're finally out of the review sections and you get to learn something new. We're finally here. It only took 7 lectures. So do we have to do the pre-class assignment before Friday? Yes, I'm gonna keep that live. We'll see if and I'll make my decision on Friday if I'm moving the Monday one back. So I wanna keep that one live because I'm an optimist, so I'm thinking we might be able to cover some of our Friday material Friday. If we don't, I'm just gonna move everything back a day. But like I said, I will let everybody know if that's happening on Friday. Luckily, if you're lost, there are resources, as in you can talk to me one-on-one. You can rewatch these videos, and you can I can give you more independent practice or find some resources for that. But yeah, right now, your goal should be start memorizing these amino acids. That should be your goal. I'll keep it simple for you. That's what you need to be focusing on right now. This PI equation is just an equation where you just have to combine 2 numbers and divide by 2. I don't think that's that big of a problem. If you want, I can give you fake problems on that, but really it is add 2 numbers, divide by 2. This is where it gets tricky. But it's not that tricky. It's just the first time you see it, your brain's gonna explode. But after it pieces itself back together, you'll find that there's beautiful logic in what I just told you after you do some practice. And it'll all make sense, I promise you. Live, lock, love. Okay. 20 amino acids. Like I said, every biochemistry student in the world, not even in America, in the world does this. I'm capable that the people in this class, if you made it through organic, this should not be that much of a struggle. Yeah, practice, right? My suggestion would be don't cram If you plan on cramming these, you're not going to learn them. So I'm trying to learn a foreign language, right? And what I do every day is I take 15 minutes to 20 minutes out of my day to study for that. And I do that multiple times a day. Overall, that adds up to an hour a day. If I tried to sit down and do an hour straight of that, I would go insane and I would hate it. But by breaking it up into 15-minute chunks multiple times a day, it's not that bad. You can do a lot of things for just 15 minutes. And that repetition is going to hammer home these structures, and they're going to start burying yourself in your head. So if you take 7 days, right, and for 7 days straight, you do 4 15 to 20 minute intervals of memorizing what an amino acid looks like, what their names are, I guarantee you're going to have it. You just have to be disciplined to do that. And that's why I suggest, if you have a smartphone, just download flashcards. This is pure memorization. Start making names for, like, to help you to remember the amino acids. Like tryptophan, when you look at the side chain, there's 2 rings. The 1-letter code for tryptophan is W. W for double rings. What's a pirate's favorite amino acid? Arginine. What's the 1-letter code of arginine? R. What's the 3-letter code of arginine? ARG. Arginine. Stupid stuff like that will help you remember the amino acids. So try to be as silly as possible. This is easier than organic. Amino acid Jeopardy game. That would be great, but we have a lot of stuff to cover, so I don't know if I can take that much time for pure memorization. So you do need to memorize the pKa's of the side chain. So there are, I want to say there's 7 side pKa's you need to know for that. 2 positive, 3 negative, and then 2 OHs. So tyrosine has a pKa, and I believe cysteine, SH, has a pKa for the side chain. Yes, I've heard that joke too, cysteine. What's a pirate's favorite amino acid? When you say arginine, you say wrong, cysteine. The cutest little amino acid that ever lived is glutamine, cute, because it's such a cutie. Look at that cutie, GLN cutie. So I made myself to be an ass on those videos so you would remember. So just again, try to be silly with it, try to have fun with it. People have told me they're crude acronyms before. If you want to be crude, if you want to be silly, whatever. People have been doing this for decades. So if you just like Google like ways to remember the amino acids, you'll probably get some ridiculous methods. But anyways, it is almost 3 o'clock. So I'm going to stop this. But if you have like questions, you want to meet with me, just shoot me an email and we can set that up. Otherwise, I hope you have a good rest of your Wednesday, and remember to breathe. That's the one thing I can tell you. If anything's ever feeling like it's bearing down on you, just some deep breaths will help you. All right. Take care, everybody.